PharmaPal.
You're listening to Bloomscast, the official podcast of Blooms.io, home of the bioactive library for mass spectrometry instant optimization. Let's dive into the latest in natural chemistry, bioactive discovery, and the future of lab innovation.
Forever, really. Humans have looked to nature for medicine, and it's, still so relevant today. Natural products are this amazing, seemingly endless source for new drug ideas.
Absolutely. I mean, think about how many treatments we rely on started from a plant or a microbe or something living.
Exactly. And that's precisely what we're digging into today.
Yeah. And what's really key here, especially, for you working hands-on in a med chem lab with natural compounds,
Right.
is getting a grip on both the incredible potential these natural products bring to the table for pharma and also the, it's very real, sometimes quite complex, hurdles in actually turning them into medicines.
Totally. We've been diving into some really good scientific articles looking at exactly that, the discovery, the development side of things. So our mission for you today is really to boil down the key takeaways. We'll look at the built-in advantages natural products often have over purely synthetic drugs.
Yeah, what makes them special.
And their huge impact, historically and now, on treating diseases. We'll even zoom in a bit on kidney diseases specifically.
That's a great example.
And then, we have to talk about the tough stuff, the practical challenges, right? Getting these things isolated, figuring out what they are, making them work better as drugs, all that.
And connecting it back, understanding these pros and cons, it's just fundamental if you're in this space. It really guides how we approach the science, what tools we need.
And ultimately, how we find those next breakthrough treatments.
Exactly.
Okay, so let's get right into it. Why the big focus on natural products versus synthetics? What makes them stand out?
Well, the first thing that just jumps out from all the research is the sheer structural diversity and the complexity. We're talking unique 3D shapes, different kinds of chemical groups attached, things that are often, really hard, sometimes basically impossible to just cook up from scratch in the lab using standard chemistry.
Yeah, these aren't simple tinker toys we're talking about. They have this kind of intricate architecture that nature's spent millions of years refining.
Precisely. And because they're so varied and complex, they cover a much wider chemical space. That's the term researchers use.
Wider than synthetic libraries, even the fancy ones.
Yeah, even compared to large collections of synthetic molecules, even those specifically designed to kind of mimic natural structures. The sources point out they often have more, say, oxygen atoms, more asymmetric centers. That's gives them that 3D bulkiness.
Okay.
And maybe fewer things like halogens or nitrogens compared to a lot of typical synthetic drugs.
So structurally, they're just playing in a different ballpark. But how does that, actually help in finding new drugs?
Well, that connects to the evolutionary aspect, this optimization idea.
Right.
These molecules didn't just appear, they evolved to do something in a biological system. Often, it's defense fighting off infections, competing with other organisms. So they've kind of been pre-screened by nature for biological activity.
Okay, and this is where the numbers get pretty convincing, isn't it?
Definitely. The stats are striking. Something like 42% of all new drugs approved over nearly 4 decades, from 81 to 2019.
Wow.
Either came straight from nature, were slightly modified versions, semi-synthetics, or used biotech based on natural templates. Compare that to only about 27% that were totally synthetic from the ground up.
So it's not just ancient history. Nature's way of designing molecules keeps hitting the mark for biological targets, maybe in ways that pure synthesis often finds harder.
That's a good way to put it, especially for really complex biological targets.
Right. And you mentioned kidney disease earlier.
Yeah.
How does this play out there? Because that seems like a really specific, important area.
It really does. And the impact there is, if anything, even clearer. Take immunosuppressants, absolutely critical drugs for kidney transplant patients, right? To stop organ rejection.
Okay, vital.
Okay, so out of the 14 approved immunosuppressants, a whopping 8, that's 57%, are either natural products themselves or very close chemical cousins, the semi-synthetics.
Over half. That's huge.
It is, and it really suggests that these natural molecules have a special knack for, interacting with the very complicated immune system, doing it effectively and relatively safely, which has been a big challenge for purely synthetic approaches in that area.
So it feels like nature just has this talent for making molecules that fit these really complex biological locks.
That's a good analogy, and many of them operate beyond Lipinski's Rule of 5.
Ah, Lipinski's rules, the drug likeness guideline.
Exactly. They help predict if a drug might work well when taken orally. Many natural products are actually bigger, maybe more complex than what those rules prefer.
But they still work.
Often, yes. Their rigidity, their defined 3D shape, can actually be a plus for hitting difficult targets like protein interactions. And surprisingly, despite being rule breakers, they're still a major source of drugs you can take by mouth.
That's interesting because you always hear that synthetic drugs are, easier to tweak to fit those rules.
And that can be true. Synthetics can often be made and modified more easily, perhaps cheaper too. And sticking to Lipinski's rules can sometimes make the development path smoother. But finding truly new kinds of active molecules within those tighter synthetic constraints, that can be really tough, especially for complex conditions like, say, Alzheimer's or other neurodegenerative diseases.
Right, so it's a trade-off. Easier development maybe versus a potentially narrower scope for finding something truly groundbreaking.
Exactly. The sources really hammer this home. Natural products are this incredibly rich, diverse pool of starting points. They cover chemical ground that synthetic libraries often miss. So for you in the lab, understanding the sheer variety is, well, it's fundamental when you're hunting for new leads.
Okay, so the advantages are pretty clear: diversity, evolutionary pre-selection, proven track record. Let's switch gears now to kidney disease specifically. You said it's a great example. Why have natural products been so impactful there?
Yeah, let's zoom in. So we know kidney diseases, things like acute kidney injury, AKI, and chronic kidney disease. CKD are major health problems worldwide.
Huge problems.
And effective treatments are desperately needed. Plus, you have the transplant situation where immunosuppressants are just non-negotiable to prevent rejection.
And this is where natural products really stepped up.
Massively. Think about drugs like tacrolimus or sirolimus. These are types of molecules called macrolide lactones, were originally found in soil bacteria.
Right, staples in immunosuppression.
Absolutely. And then you have their slightly modified cousins, like everolimus, temsirolimus, also widely used. These all came from nature's toolkit.
And these aren't just minor players, are they? They're frontline treatments.
Totally. The research points out if you look at the top 10 drugs used for immunosuppression and preventing infections in kidney patients, a remarkable 7 out of those 10 are either natural products or directly inspired by them.
7 out of 10. Wow.
Yeah. Now, sure, there are important synthetic drugs too, like benazepril or valsartan used for diabetic kidney disease, but they don't really change the overall picture. Nature-derived molecules are dominant, especially in that critical immunosuppression space.
It seems like those features we talked about, the diversity, the bigger size, the 3D complexity, the stereo centers, they must be particularly useful for tackling kidney disease issues.
That's exactly what the evidence suggests. The natural products used successfully in kidney treatments often do have those characteristics. And the conclusion from the sources is pretty bold. The importance of nature-derived drugs might actually be even higher in kidney disease therapy than in drug discovery overall.
That's a really strong statement. Okay, so the potential is undeniable. But like anything this good, there have to be catches, right? Big challenges in actually getting these things from nature into a medicine bottle.
You absolutely hit it. That's the flip side. The very things that make them amazing, their complexity, their natural source, also create some pretty significant hurdles.
Okay, let's start at the very beginning, just finding them and getting enough to even study. Isolation. What are the roadblocks there?
Well, the traditional way, this bioactivity-guided isolation from crude extracts, it's often described as, frankly, laborious. Really time-consuming and tricky.
Yeah, I can imagine. Sifting through goo.
Kind of. And a huge issue is just growing the source organism. A lot of these microbes or plants, they don't like lab conditions.
Right.
You take them out of their natural niche, and maybe they stop making the interesting compound altogether, or they just don't grow well. I remember spending ages on a plant extract once.
Oh, yeah.
Trying to purify one specific thing. It felt like chasing a ghost, tiny, tiny amounts, and separating it from all the other stuff was a nightmare.
Like trying to grow a rare orchid in your backyard when it only lives on one specific mountaintop. It just might not work.
That's a great analogy. And even if you can grow it, the amount of the actual bioactive compound you get might be minuscule. Just not enough to do all the tests you need, let alone think about scaling up.
Okay, so getting it out is hard. Then comes figuring out what it is you've actually got. Identification challenges.
Yeah, that's the next big one. These extracts are, complex mixtures. hundreds, thousands of different molecules maybe. Finding the active one in that chemical haystack is tough. And this is where dereplication comes in. It's crucial. It basically means trying really hard not to rediscover something we already know about, because that's a huge waste of time and money.
You don't want to spend 6 months isolating aspirin again.
Exactly. But dereplication itself has limits. Plus, sometimes those crude extracts just don't play nice with modern screening methods, like high-throughput assays looking for specific targets.
So the analysis is tricky. What tools do you use?
We rely heavily on things like metabolomics, NMR, that's nuclear magnetic resonance and high-resolution mass spec, HRMS. Powerful tools for looking at molecular structures.
They're not perfect.
Not perfect. They have limits on sensitivity. Sometimes the data is incredibly complex to interpret, and nailing down the exact structure of every single thing in a mix, still very challenging.
And just keeping track of all the known natural products must be a job in itself. There must be thousands upon thousands.
Oh, hundreds of thousands, easily. Databases like the Dictionary of Natural Products, or platforms like GNPS, the Global Natural Products Social Molecular Networking Platform, are absolutely essential for dereplication.
GNPS. How does that work?
It helps visualize relationships between molecules based on their mass spec data. Really clever. But even these amazing resources aren't totally complete, and you still need a lot of expertise to interpret the results correctly. People are constantly working on better computational tools and bigger spectral libraries to improve this.
Okay, so say you manage the isolation and the identification.
Yeah.
You've got your molecule. Is it usually ready to go as a drug straight away?
Almost never. That'd be nice, wouldn't it?
Yeah.
But no. Often, the natural product, even if it's really potent in a lab test, might not have the right drug-like properties. Meaning things like, how well does it get absorbed if you swallow it? How does the body break it down? Does it have toxic side effects? That sort of thing.
So it might work great in a dish, but be useless or even harmful in a person.
Exactly. And that leads to the next big challenge. Enrichment, or optimization. You usually need to make slightly different versions, structural analogs of the natural product.
To try and improve it, make it safer, more effective.
Precisely. Tweak the structure to boost the good properties and reduce the bad ones. But here's the kicker. If the original natural product is super complex and hard to make synthetically, then making those tweaked versions for testing for structure-activity relationship studies becomes incredibly difficult, maybe even impossible.
That makes sense. If you can't easily fiddle with a structure, you're stuck with what nature gave you, warts and all.
Pretty much. It really limits your ability to optimize it into a proper drug.
Now, something you touched on with the source organism's variability. How do you ensure consistency? Quality control must be a headache if the source material itself changes depending on where or when you collect it.
That's a really sharp point, and while the papers we looked at didn't dedicate a whole section to it, it's a huge practical issue. You're right. Where a plant grew, the season, the soil, the weather.
Or the specific strain of bacteria.
Exactly. All these things can change the chemical profile of the extract. So ensuring that batch number 100 is the same as batch number 1 in terms of the active compound and potential impurities, that adds a massive layer of complexity to quality control for natural product-derived drugs.
Okay, another hurdle. What about figuring out how the drug actually works? Its mechanism of action.
Yeah, that can be another bottleneck. Often you find these things using phenotypic screens.
Meaning you see an effect like it kills cancer cells, but you don't know how.
Exactly. You see the outcome, but the specific molecular target or pathway it's hitting, that can take a lot more work to figure out. It can be really time-consuming.
But I guess science is getting better at that, too.
Oh, definitely. The sources do mention big advances here. New screening methods using things like induced pluripotent stem cells or gene editing like CRISPR, and also better techniques specifically designed to find a drug's target methods with acronyms like NTOT, DARTS, CETSA. These are definitely helping to speed up figuring out the how.
Right. Okay, last hurdle area, maybe. The practical stuff, patents, regulations, getting enough material long term.
Yeah, the sort of business end challenges, but absolutely critical. Intellectual property, IP, is a big one.
How so?
Well, getting a strong patent on a natural product exactly as it exists in nature can be tricky. The rules vary, but generally just finding something natural isn't always seen as inventive enough for a patent in its original form.
But modifying it helps.
Yes. Often even small chemical modifications can create a new patentable molecule. That's a big driver for making those semi-synthetic versions we talked about.
Okay. What else on the regulatory front?
Benefit sharing. This is huge now. International agreements like the Convention on Biological Diversity and the Nagoya Protocol.
Right, ensuring the source country benefits if a drug is developed from their resources.
Exactly. It's about fairness, but navigating those rules, ensuring compliance, adds complexity and cost. And it's increasingly relevant for marine resources, too.
So more layers of administration and legal stuff. And finally, just getting enough of the raw material. Sustainably.
You nailed it. Sustainable supply. If your wonder drug comes from a rare orchid that only grows on one cliff face.
Yeah, that's not going to work for mass production.
Right. Or even if it's a microbe, maybe it grows incredibly slowly or needs very specific, hard to replicate conditions. Ensuring you can get enough material consistently and sustainably for research and potential manufacturing, that's a fundamental challenge you have to solve early on.
Wow. Okay, so it definitely sounds like the path from finding something cool in nature to getting it into a pharmacy is uniquely challenging for natural products.
It really presents its own distinct set of often complex obstacles, absolutely. But as we said, the potential payoff finding truly novel drugs is enormous. And it's worth remembering the field isn't standing still. New technologies, smarter approaches are constantly being developed to tackle these very challenges.
Right. So let's try and wrap this up. We've seen this incredible value in natural products, an amazing structural diversity, the evolutionary head start for biological activity, making them this super rich hunting ground for new drugs. And we saw how that plays out powerfully in areas like kidney disease treatment.
Absolutely spot on. But, and it's a big but, it's just as vital to appreciate those significant multifaceted challenges. They pop up all along the way, right? From just finding and isolating this stuff to figuring out what it is, making it more drug-like, dealing with quality control, understanding how it works, and then navigating all the IP and regulatory hoops, not to mention securing a reliable supply. It's a lot.
It really is. Okay, so here's something to chew on, especially for you listening who might be working in this area. We know natural products offer this unique chemical space, this deep biological relevance. So the big question is, how do we as medicinal chemists, as drug developers, best bridge that gap? How do we really harness nature's ingenuity, overcome these inherent challenges, and integrate those principles effectively with the demands of modern drug development to really unlock that next wave of life-saving medicines?
That's the million-dollar question, isn't it? Finding that perfect balance.
Definitely something to think about.
Thanks for tuning in to Bloom's Cast, brought to you by blooms.io. Stay curious, stay connected, and we'll see you next time on the frontier of natural product innovation.
PharmaPal.